Hello, everyone. Thank you for attending the workshop today, which is focused on replacing regulatory experiments on animals, an introduction to in silico models and in vitro test methods. My name is Emilia Kostin. I work as a manager for scientific services and study director at the Institute for In Vitro Sciences. I've been at IVS for 10 years now. Before launching into my presentation, which is going to be concentrated on safety assessment for skin, particularly irritation and corrosion, I would like to share a few words about my scientific journey, which may be relevant to your group. Looking backwards to my career, I think I was blessed to understand how the process to translate an idea into a labeled product. I started my PhD years working for the Institute of Biochemistry of the Romanian Academy in Bucharest, Romania. And I did my postdoc years at the National Institute of Health, National Cancer Institute, and I worked a lot with various chemicals and their impact on various cell lines, particularly melanocytes that are the pigment-producing cells in the skin. After finishing my postdoc years, I transferred to industry, to a large manufacturing company, Avon Products, where I had the view of the process that launches products on the market for personal care and cosmetic industry. After working for Avon for a little while, I started to work for the Institute for In Vitro Sciences, also part of the industry, but a different view from the contract research organization standpoint. So over almost 20 years of my career, I transferred from biochemistry to cell biology to currently to toxicology. I'm a biochemist by formation, and I became a toxicologist recently, and I am working in a very exciting area of in vitro toxicology, which is focused on replacement, reduction, and refinement to replace animal studies that are used for safety assessment of products. At IVS, we follow a global approach to training. Our activity today is part of that mission that we have. And colored on the world map are the countries where either we went to provide training in vitro methods or countries from which representatives came to our institute to be trained in vitro methods. And as you can see, we have been pretty much all over the world, and we hosted scientists from regulatory groups, from academia, from industry in our facility. We have been recognized for our efforts in training by being awarded twice the LASH Award, first time in 2012 for training, and in 2016 for the Young Researcher Award, which was presented to one of our colleagues, Dr. Kim Norman. For activities related to colleges and students, I should point out that we hosted 3 or more workshops at our facility for a group at University of Maryland, the Bioengineering Society workshop, and this is a picture showing the activity in our lab. My presentation will focus on an overarching view of our activities, and I will share on perspective, challenges, common goals, and how we can all work together with the same goal in mind, and that is the safety of the consumer or the safety of the end user of a product. If we start our journey today through my presentation from the idea that a manufacturer, any company in industry has an idea and has a prototype of a product that is unlabeled and uncharacterized, this product has to be tested using preclinical testing, either classically in vivo using animals or in vitro using various methods that we are performing at IVS. The data eventually are submitted to a regulatory agency, and I have listed here the United States Environmental Protection Agency, or US EPA, only because we worked with the agency closely in recent years, but it could be any relevant regulatory agency. After the review at the agency, all the data that are collectively analyzed will ensure that the consumer or the user doesn't have any adverse effect on the skin, like in this picture. We will go through an in-chemical assay. We will follow with more complex in vitro assays that are either cell-based or based on reconstructed tissues. And we will discuss 2 modern concepts that are critical to today's toxicology. One is peer testing, and the other one is integrated approaches to testing and assessment, or IATAs. Finally, we will discuss in vitro assays and emerging technologies like organ on a chip or human on a chip. It can be seen in this slide, the in vitro methods are we are fairly demanding on them, and we are what I call a demanding customer because the in vitro assays should be high throughput, specific, sensitive, validated, and reliable, reproducible, relevant, and so on. So it's quite a lot that gets into performing and perfecting an in vitro assay that is capable to provide the same prediction as the animal assays which are classically used for the safety assessment. I would like to touch upon the reductionist concept of in vitro models, not related to skin. However, the concept is valid for all endpoints. And here is what I call less is more. Over the years, the methods that have been used to assess safety of humans have become smaller in size to address specific endpoints. If in the 40s, Draize designed the animal test using rabbits, over the years in the 90s, the test system became smaller but more targeted to the endpoints of interest, either using organs like the eyeball in my slide or resected bovine corneas or cell cultures from rabbits. The test system became smaller and smaller and more targeted. We are now in a different era in the 2000s and on, where the complexity of the in vitro systems, as small as they are, is increased. As you can see in the past, several lines or several organs from different species have been used to assess safety, whereas right now the test systems are using human cells or human-derived tissues so that the test system is actually relevant to a human exposure. And the science has evolved tremendously from cell culture to using reconstructed tissue models that I'm going to discuss about, organs on a chip like in human retina here, or even body on a chip, as I will present a little later. In the cycle of producing a product or manufacturing a product, either a chemical or a cosmetic product or a household cleaner, everything starts with a concept or with an idea. The product, the idea is converted into a prototype that needs to be tested for safety and efficacy. There are a couple of assays that can be used for this purpose, either simpler in silico or in chemical assays. Since we're talking about skin, which is the largest organ of our body and very complex for that matter, I look at skin with its multi-layer as a very deep type of tissue. So I look at investigating skin like to a journey. So one needs to go skin deep and understand all the mechanisms that are involved in transforming a toxicant byproduct eventually less toxic. What we can use in this situation going through the skin, we can use either native skin like explants or reconstructed tissue models. We can go even a little deeper and use specific cells to the skin, like keratinocytes or melanocytes, either as simple cultures or co-cultures, and we can perform in vitro methods. One can even go deeper than that using organelles or proteomic analysis. Eventually, after going that deep into understanding the effect of toxicants on skin, one needs to come back to the surface in like a journey to the surface and understand how a product impacted the human skin. If the product is safe, then the consumer will have absolutely no reaction to the toxicant. And in this way, our unlabeled, uncharacterized product is finally getting a label and all the safety assessments that are necessary before reaching the consumer. As I said, human skin is a very complex tissue or organ that represents an immune network and a defense system against UV, against mechanical, chemical, and microbial factors. When a toxicant is applied to the skin, either corrosive or an irritant, it can induce several reactions. One of them is corrosion, and the other one is irritation that we're going to discuss today. Corrosion is an irreversible damage of the skin following the exposure to a test substance, and it's characterized by ulcers and bleeding, as shown in this picture. Irritation is a reversible damage of the skin following exposure to a test substance and is characterized by erythema, the redness, and edema. Eventually, skin can recover after an irritating event. It is, however, important for companies and for consumers to see the appropriate labels on the product that they come in contact with, either purposely or accidentally. Therefore, there are a couple of methods that are used for the registration and labeling of chemicals, transport of chemicals, for occupational safety, and for safety of cosmetics and household products and any other products that are put on the market. In the past, historically, an animal test system was used to address the acute dermal corrosion and irritation assessment, and it was called Draize test. The test used albino rabbits, and the endpoints of the assay were erythema and edema. The assay applicable to evaluate the corrosion and irritation potential of test substances, and the results can be submitted to a regulatory agency like US EPA, for example. However, like any assay, there were limitations in the sense that the rabbit and the human skin have different physiological properties. Therefore, they have different responses to environmental and chemical agents. Some of the compounds could be more toxic to rabbits than humans, and vice versa. Furthermore, a dated ethical issue of the in vivo test concerns to animal suffering and discomfort motivated the scientific community to move towards the next generation of tests, namely in vitro assays that would eventually replace the animal test. The Draize test has an associated OECD test guideline, where OECD is the Organization for Economic Cooperation and Development, responsible for the vast majority of the validated studies. In brief, the Draize test uses 1 to 3 rabbits. In the far past, the assay was performed even with 6 rabbits at the time. The exposure time are relatively short for the corrosion assay, anywhere from 3 minutes to 1 hour or 4, and longer for skin irritation up to 4 hours. There is a reason for that, because corrosive materials are very strong penetrators of the skin, and they create a lot of toxicity, induce a lot of toxicity in a very short time. The animals are observed up to 28 days, and individual scores and total scores of the erythema and edema endpoints are calculated. The animal test is capable to cover a wide range, a continuous range of irritation potentials, anywhere from corrosive materials to basically non-irritant materials. In order to assess this wide continuum of irritation potential in vitro, several assays have been designed over the years. One of the assays that I would like to discuss today is an in-chemical assay based on the membrane barrier test method called Corrositex. The assay uses an artificial membrane that is designed to respond to corrosive substances in a manner similar to the animal skin. The assay endpoint is represented by the minutes, the time in minutes required for a test substance to penetrate through the Corrositex biobarrier membrane and to produce a color change in the chemical detection system. All the assays that are performed use specific assay controls, negative and positive, similarly to the animal model. This particular in vitro assay is used to assign UN packing groups to corrosive materials or to verify if a test substance is non-corrosive. One of the limitations of the assay resides with the pH values of the test substances to be assessed. Usually, materials with a pH between 4.5 and 8.5 do not qualify for testing based on the chemical detection system used in the kit, in the sense that they do not induce a color change visible to the assessor. The assay is validated for regulatory purposes, and the OECD guideline is 430. In brief, this is how the assay works. The biobarrier membranes are prepared from a powder that is provided in the kit. The matrix is added to a membrane disc containing cell membrane, and it's placed on the vial containing the chemical detection system. Each test substance is added then to 4 replicas by barrier membranes, and the vials are continuously monitored for any color change for the first 10 minutes until a breakthrough occurs. When the first color change is observed, this is the so-called breakthrough time. The in vitro assays use prediction models where the endpoints are used to predict either packing groups, like in this case, or other classifications compared to the animal test. The Corrositex assay is capable to categorize the corrosive materials and to assign specific packing groups, which are extremely important for the transportation of substances, particularly corrosive, and for worker safety. The assay is used particularly by the Department of Transportation in the United States. Moving into more complex in vitro assays, I would like to emphasize several test systems that can be used to assess the safety of the skin. One of them is based on conventional cell cultures. They have their own advantages and their own limitations. One of the advantages is the production of large number of cells and the possibility to self-culture and cryopreserve for a long time. Under the limitations, the cells, since they are simple cell cultures, the maintenance of all differentiation characteristics is questionable, and they do not have the stratum corneum, which may result in high sensitivity and over-prediction of irritation potential. Promising and evolving rapidly are the epidermal equivalents or reconstructed tissue models, which have a series of advantages. They present the penetration barrier in the form of the stratum corneum, and they offer the possibility to test products that are water insoluble, such as final formulations, like lotions or creams. Finally, skin explants can be used from surgery, for example, and they have similar advantages to the epidermal equivalent. One of the limitations, however, is the short time experiments that can be performed with the skin explants after being rejected from the human body. We will concentrate today on the use of reconstructed tissue models. When designing various tissue models, one can think of the routes of exposure that we discussed earlier. And I will use an example of different tissue models from the US manufacturer Matek Corporation. For example, for the route of inhalation, the company generated a model called Epi-Airway. For ingestion, there is a specific tissue model called epi-intestinal. And for skin, since this is the topic of my lecture today, they have a product that is called the Epiderm. The use of reconstructed tissue models is very important, particularly before any human clinical studies, should we talk about pharmaceutical compounds. They play a very important role in saving a lot of time and a lot of money up to the clinical trials, because they are capable to screen an increasing number of compounds from a library, for example. As I said before, native human skin is a very complex organ, and it has multiple layers, stratum corneum, and a couple of others underneath that provide the safety of our body. Multiple reconstructed tissue models that are now validated for regulatory purposes have been produced by different producers around the world, as shown in my slide. All these models are generated from human keratinocytes, and they show multiple layers of viable epithelial cells. Most importantly, the stratum corneum is functional and it offers a barrier that is specific to the skin. When received at the end user, the tissues are received in small inserts that are called cell culture inserts, and the tissue is reconstructed on the top of membrane that is capable to absorb the media from underneath the tissue. This is an example of one of the models, specifically Epiderm model from Matek Corporation. The reconstructed skin models can be used for a couple of purposes. One of them is for healing, for burns and non-healing ulcers, for example. In this slide, you can see one example like that, which is the strata graft. it is produced using unmodified mixed human keratinocyte progenitor line. This line has been originally isolated from a normal human infant tissue, and the line spontaneously became a continuous cell line that can be grown indefinitely. This is a product from Stratatex Corporation, and this picture shows the performance of the model when grafted on a patient after 3 months compared to an autograft, which is basically skin from the same patient but from a different area of his or her body. The reconstructed skin models can be also used for dermal safety assessments, the focus of my presentation today. The first assay that I'm going to discuss is the skin corrosion assay, which is covered by OECD test guideline 431. There are 4 tissue models that are validated for regulatory purposes as listed in my slide: Epiderm, Episkin, Kinetics, and EpiCF. The assay endpoint is represented by the tissue viability assessed by the reduction of the vital dye MTT. I will explain the mechanism of MTT in the next slide. The assay controls are represented by the negative control, which could be sterile deionized water or sodium chloride, and the positive control, which is abnormal KOH or glacial acetic acid. The results of the study can be used for regulatory purposes to distinguish corrosive materials from non-corrosive materials. The assay also is used to subcategorize corrosive materials from category 1A versus categories 1B and 1C together. The method as a limitation is not capable to discriminate yet between category 1B and 1C corrosive materials due to a limited set of well-known in vivo corrosive materials from category 1C. I would like to say a few words about the mechanism of MTT reduction as the measure of cells or tissue viability. MTT is it's a dye. Originally, it's of yellow color, as shown in my slide, which is transformed in the mitochondria of viable cells, metabolically active, into its reduced form, which is the formazan, and it is a purple, dark blue color. And as you can see, these are the groups, the chemical groups, that the chemical bond that is basically broken to give a different color when the compound is actually metabolized. And this is how the samples look like in the laboratory, where a viable tissue would basically reduce the MTT to its formazan form, and the dead tissue that is not metabolically active will not be capable to induce the same color change. This is how the typical corrosion protocol is performed. After the tissues are received in the lab, they are incubated for at least 1 hour in their culture medium. Then the media is refreshed, and the tissues are redosed with the test substance of interest for the specific time, in this case 3 minutes or 1 and 4 or 4 hours. After the completion of the exposure, the tissues are rinsed with usually DPBS. They are incubated for 3 hours in MTT to allow the reduction of MTT. The MTT is then extracted in isopropanol for 2 hours. And the extracted MTT is sampled into a 96 well plate, and the optical density is read, and the optical density is converted then into viability values of the tissues. Based on the viability values calculated for the tissues and specific cutoff values for each validated tissue model, this in vitro assay is capable to separate corrosive materials, subcategory 1A, which is the most aggressive, from corrosive materials as a combination of 1B and 1C subcategories, and non-corrosive materials. The next assay that I would like to discuss is the skin irritation test, based on, again, reconstructed tissue models validated for regulatory purposes, as listed on my slide. The concept is fairly similar, using the tissue viability assessed by the MTT endpoint, and the assay is using negative and positive controls. The assay is applicable in separating category 2 or irritant materials from non-irritants for skin using the globally harmonized system. This is how the assay is performed, similar to the corrosion. However, there are a couple of differences. First of all, the tissues are pre-incubated for 1 hour and then overnight before being treated with the test substance. We use triplicate tissues. And the tissues are post-incubated for a total of 42 hours to capture delayed effects of skin irritants. The same steps are following in using the MTT reduction, isopropanol extraction, and sampling out the extracted MTT to calculate the viability of the tissues. The prediction model uses a cutoff value of 50% of the tissue viability, where products that reduce the viability of the tissues to less than 50% are considered category 1 or 2, in this case, GHS2, and vice versa. If the tissue viability was more than 50%, the products are predicted to be non-irritant. I included in my slide the link to a video that shows the skin irritation test as performed using the Matec Corporation tissue model Epidermis. In the interest of time, and since the video is fairly long, I will not launch it through my slides, but feel free to visit the website that is listed on my slide and watch it if you're interested. Since we talked about skin corrosion and skin irritation, I would like to present to you the testing strategies that use the two endpoints. If a product is considered to be potentially corrosive based on pH values or based on other data that a manufacturer may have, we can perform the skin corrosion assay, the in vitro assay in our lab. If the products are predicted to be corrosive, then the product can be labeled as such. And this is what we call top-down strategy. If the products are predicted to be non-corrosive, we do not know if they are irritants. So we could follow the so-called bottom-up strategy, where one presumes that a product is irritant, but not corrosive. We perform the skin irritation test. If the product is predicted to be an irritant, and if we started from this end of the strategy, then we have to assess if it is as irritant as a corrosive material, and therefore perform the in vitro corrosion assay. And if the product is predicted to be non-corrosive, then it can be labeled as such. After we discussed this pure testing strategy, I announced at the beginning of my slides that we're going to briefly touch upon the IATAs. They are a very modern concept in toxicology, and they use a way of evidence, collective data, either animal or in vitro data, physical chemical properties, when making an assessment for the safety of test substances. If testing is necessary, here is the area where the in vitro assays can fit in, particularly for the skin corrosion and irritation. If you're interested to learn more about the IATAs, here is the link that you can visit. So let's get back to the Draize continuum of irritation and corrosion. You remember that the animal test was capable to cover all this continuum, and the in vitro assays can do the same. However, since the Draize test, you remember, would follow the animal up to 28 days, it is rather difficult to fit into in vitro assays in a very short time. So a battery of assays are actually used to cover this continuum of irritation and to assess the safety of a wide variety of products, from industrial chemicals to color cosmetics. So we're working with a fairly short timeline in vitro, from 3 minutes to 24 hours. So for corrosive materials, we can use the in vitro skin corrosion assay. For severe irritants or non-irritants, we can use the validated assay skin irritation test. As you can see, these assays provide a yes or no type of answer. And this is what the regulatory agencies are usually interested. In the middle of the continuum, we have a multitude of in vitro assays that are non-regulatory, but that can assess moderate to mild to non-irritant potential. And if we want to put this irritation continuum on a scale of MTT reduction, you can see how corrosive materials do not reduce the MTT, and non-irritant materials are not affecting the MTT, which is reduced by metabolically active cells. We talked about what is currently used to assess the safety of skin, and you may wonder what future holds. Whether we're working in pharmaceutical industry, cosmetic industry, or household cleaning products industry, we're moving towards high throughput assays, either based on bioprinting, or we're moving towards an all-inclusive human model concept, or human on a chip. If we're thinking back how the computers were a while back, we can remember the hard drives, which were relatively large, and they were holding some information, but not as much as the size of the drive. And today, we're stocking probably the same amount of information, if not more, on a very small flash drive. So it's the same in science. So one example of this is the concept of bioprinting. And I would like to launch now a video from the company Organovo. Credit to the company, where you will see how bioprinting works.
Organovo creates functional human tissues with its unique bioprinting technology. Starting with cells from any source, Organovo's bioprinter precisely places a specially formulated bioink, in this case, into a 96-well plate for drug discovery. The process results in a complex tissue with natural architecture. Disease can be induced, and disease progression can be studied in the native microenvironment. For medical research and drug discovery, potential treatments can be observed real-time to measure safety, efficacy, and mechanism of action. 3D bioprinting can create more natural cell-cell interactions than traditional 2D methods, with better predictive value and higher clinical relevance. This revolutionary technology can speed safe and effective drug development, providing an entirely new way to study disease and advance medical research, and ultimately create a potential new source of tissues for surgery and transplant medicine. Organovo, changing the shape of medical research and practice.
Back to our presentation, let's go back to the concept we started the lecture with: perspectives, challenges, common goals, and working together. Remember the prototype we started with that was unlabeled. We managed to get it labeled, appropriately assess for its safety, put on the market, and ensure that no adverse effects will show up on the skin of the humans that are using it. The industry manufactured the product. However, other forces came together, like trade associations, animal welfare groups, and academia, to make this possible. Another player in this perspective are the safety testing labs, like IIVS, which perform the assays, which are sensitive, high throughput, reproducible, and specific, and provide reliable data for the safety of humans. Also, the labeling and regulatory agencies are working hard to ensure that the field is moving towards animal replacement, and these are a couple of key documents and regulatory laws that were issued by the US EPA to support this particular endeavor. After hearing about the safety assessment of skin, you may wonder, where does your field and expertise fit? I would say in all these areas. If you are in this program, you can probably have a job either in research, consulting, education, industry, animal welfare, and so on. So pretty much the answer to my question is everywhere. You may wonder, though, why is a chameleon on my slide? I use this concept because I heard it the first time a couple of years back from Dr. Milinkovic, who is a professor at University of Geneva. And he emphasized in his lecture about the color of reptiles, about the key of working in a multidisciplinary team. Being in a biological field, you can fit in a multidisciplinary team that can work in toxicology or in any other field, and to become the next generation of toxicologists if you wish to. As for acknowledgments, I would like to bring back field we work in vitro toxicology, moving towards replacement, reduction, and refinement, and today, a couple of players came together and worked together for this presentation to happen. And those are the Institute for In Vitro Sciences, PETA, US EPA, who supported multiple validations, and your university being such a good host to us. Thank you very much for your attention.